Hello, hi viewers, welcome to my channel. Today we will discuss about one of the most interesting topic, that is respiratory quotient. This topic will be very helpful in bioprocess engineering and technology, as well as it will be very useful to aspirants who are looking for GATE examination and TANCET examination in biotechnology, and also it will be useful for NEET examinations. Let's move on to the topic. The definition of respiratory quotient. It is defined as the moles of carbon dioxide produced per moles of oxygen consumed. The formula to calculate respiratory quotient is RQ is equal to moles of carbon dioxide produced divided by moles of oxygen consumed. And we will look into the uses of respiratory quotient. The first use is it is used as an indicator for the metabolic rate, in which the metabolism per unit time can be measured based on consumption of food or the energy released in the form of heat or how much amount of oxygen is being used for metabolism. And the second use is it represents the oxidation reaction of different substrates. This will give us an idea about how the different substrates are being used for metabolic process. And this respiratory quotient value will vary depends upon the substrate which we are used for respiration. You can see the RQ value is being varied in terms of humans and animals. So considering the human, the RQ value will vary depends upon the substrate which it is being metabolized. Okay, for fat it will be of zero point 0.7, and for protein it will be of zero point 0.8, and for carbohydrates it will be of 1. And coming to the animals, when the animal is at rest, the RQ value will be of zero point 0.8 to zero point 0.9. This table will illustrate the RQ value of different substrates. For fat, the RQ value will be less than 1, and this RQ value for each type of fat may vary. Okay, and coming to the protein, the RQ value will be of zero point 0.8, and for carbohydrate it will be of equal to 1. Here comes the term respiration. It is the metabolic process that takes place inside the cell of all living organisms, in which energy will be released on breakdown of substrates, and this respiration will be categorized into two forms as aerobic respiration and the other one is anaerobic respiration. Let's look into the difference between aerobic and anaerobic respiration. And this aerobic respiration will take place in the presence of oxygen. Anaerobic respiration will take place in the absence of oxygen. And we can see aerobic respiration in higher organisms like plants and animals. We can see anaerobic respiration in human muscle cells, algae, bacteria, and yeast. And the end product of aerobic respiration will be of carbon dioxide and water. And in case of anaerobic respiration, the end product will be of alcohol. And the major difference is the amount of energy that is produced between aerobic and anaerobic respiration is, in case of aerobic respiration, the energy release is very high when compared to anaerobic respiration. Okay, so now we will look into the problem to find the respiratory quotient in terms of aerobic and anaerobic respiration by considering the glucose as an substrate. So you can look into both the equations. So first we will solve on aerobic respiration. So here they have given the complete oxidation of glucose molecule. So you can see here how the glucose is being utilized as a substrate, and the product, that is carbon dioxide and water molecule, is being generated. Okay, and we all know that the formula for RQ is moles of carbon dioxide produced divided by moles of oxygen consumed. Okay, so now in this equation you have to look into the carbon dioxide term and oxygen term. So here 6 moles of carbon dioxide is being generated by consuming 6 moles of oxygen. So therefore the RQ value will be of 1. So in the same way we will calculate the RQ value of glucose in anaerobic respiration. So the uh, major difference between the aerobic and anaerobic, as I told that it will take place only in the absence of oxygen. So therefore here there will be no oxygen molecules. Okay, so therefore we will calculate the RQ as 2 moles of carbon dioxide divided by 0 moles of oxygen, so which will lead to infinity value. Okay, so you may ask why the RQ value during anaerobic respiration is infinity. As we all know the difference between aerobic and anaerobic respiration, in case of anaerobic respiration it will take place without the involvement of oxygen, so that's why the respiratory quotient value will be infinity. Okay, so based upon the respiration quotient value we can tell whether the respiration is of aerobic form or anaerobic form. If the respiratory quotient value will be of equal to 1 or less than 1, then we can say that the respiration is of aerobic. If the respiratory quotient value will be of greater than 1, then we can surely say that the respiration is of anaerobic respiration. So now this problem was asked in the GATE examination by technology in the year 2014. The most plausible explanation for a sudden increase of the respiratory quotient of a microbial culture is that option A, cells are dying, option B, yield of biomass is increasing, option C, the fermentation rate is increasing relative to respiration rate, option D, the maintenance rate is decreasing. So from that we have to select the correct answer. So I will give you the solution. So we all know that the growth of bacteria or the microbe will occur in different phases. When the bacteria is growing rapidly, it required high level of dissolved oxygen. So at that condition, the microbial metabolism is being controlled on the basis of respiratory quotient. So from the given option, the correct answer will be of the fermentation rate is increasing relative to respiration rate, that is option C. Thank you for watching.